Good afternoon. The Recursion team is here today to talk about our high-dimensional platform and capabilities for accelerating drug discovery. I'm Blucher, and I'm a senior data scientist, part of a team that works on translating our platform discoveries to really focus questions about druggability and therapy development. So disclaimers. Speakers you're going to hear from today are employees of Recursion, and the views and opinions in this presentation are their own and not necessarily those of Recursion Pharmaceuticals. Their accuracy and reliability are not guaranteed. The material presented is general background in summary form and may not be current or complete. This presentation is intended for educational purposes only. Here at Recursion, our mission is to decode biology to radically improve lives. As part of that mission, we generate over 1.3 million cellular images like the ones you see above, and we do that every week. We've generated more than 9 petabytes to date. Today, our team is here to tell you about what we do with these data. You're going to hear from a few different folks who interact with the data at different levels, and this is an intentional choice because we're working on hard problems that require cross-functional teams.
Hi, my name is Berton Earnshaw, and I'm a machine learning fellow at Recursion. I'm responsible for Recursion's machine learning strategy. I will now describe Recursion's high-throughput imaging platform for generating high-dimensional, biologically relevant phenotypic readouts, and how we create useful representations of those readouts. These representations power much of Recursion's drug discovery engine. We designed our phenomics platform to model a wide range of biology spanning multiple therapeutic areas, including oncology, immunology, neuroscience, cardiovascular, metabolic, and infectious diseases, using the same image-based fluorescent microscopy readout. The platform's design enables us to systematically expand our search space into new areas of exploration while minimizing the need for bespoke assay development. Using a consistent phenomic protocol enables us to analyze and compare the resulting data across a wide variety of conditions, revealing the interconnectedness of human biology and tractable therapeutic starting points. For example, to understand the effects of gene deficiency, we knock out each gene in the human genome in disease-relevant cell types using CRISPR gene editing in an arrayed, high-throughput format. Guide sequences are designed in-house to be more efficient. Images of these genetically perturbed cells are then featurized using a neural network that is trained to capture the wide array of morphological characteristics these cells exhibit. The result is that each image is transformed into a vector of numbers that represents the morphological characteristics of the cells in the image. Similar representations can be obtained on the phenomic platform by dosing cells with small molecules from Recursion's libraries of known and new chemical entities. The resulting vector representations of gene deficiency and molecular perturbation can then be analyzed together in the same vector space in order to map out gene-gene and gene-compound relationships, including protein complex membership, pathway regulation, and target identification. You will see some examples of these applications later in this talk. This and related CRISPR tools enable us to broadly interrogate human genetics, ranging from Mendelian genetic diseases to precision therapeutics. Moreover, we can use our phenomic platform to interrogate other families of diseases, including those induced by soluble factors and infectious agents, as well as those modeled by co-cultured cells or patient-derived iPSCs, and many others. Many high-throughput screening technologies can screen either a large number of genotypes or a large number of compounds, but not both. In contrast, Recursion's phenomic platform is capable of efficiently producing relatable data across many different perturbation types, including genetic and small molecule. Moreover, the screening can be done in a variety of cellular contexts, including different cell types or induced states, for example, TNF-alpha-induced inflammation, ensuring biologically relevant readouts. Recursion has generated more than 9 petabytes of phenomic data, spanning dozens of cellular contexts and hundreds of thousands of genetic and chemical perturbations to those contexts. This dataset contains a massive amount of cellular morphological variation. In order to properly capture the biologically relevant morphological characteristics of each cellular image, we train neural networks to perform a variety of tasks with these images and use intermediate layers from those models to provide embeddings or representations of those images. Training perturbation classifiers on well-chosen subsets of this data yields models that not only generalize extremely well out of batch, they produce biologically relevant features that perform well in many downstream drug discovery tasks. How to best learn representations of our phenomic data is an ongoing research topic of primary importance at Recursion.
Hi everyone, I'm Safiye Celik. I am a data science manager on the Recursion team. I will now tell you about some of the results from our benchmarking of the learned embedding space. As Berton talked about earlier, this embedding space is learned by our deep learning pipeline run on images from our in-house compound and CRISPR screens. Here is the pairwise similarity matrix for genes annotated in a diverse set of biological pathways. The similarity value here for a gene pair is the cosine similarity between the two genes based on their representation in the high-dimensional embedding space. As can be seen by the dark red squares at the diagonal of this heat map, the knockout of genes that are in the same pathway are more similar to each other than the genes in other pathways. For example, the very tight cluster on the upper left, labeled by yellow, represents the proteasome numbers. We would expect here the gene knockout to result in highly similar phenotypic effects due to the disruption of the proteasome complex. And the two clusters at the bottom right, labeled by red and orange, represent exosome and ribosomal subunit. And in addition to having two tight clusters for each of these gene sets, we also see a larger shared cluster across the two. And this points to non-collaborative functionality between these biological processes. Just as we looked at clusters of genes in the previous slide, we are looking here at how well we can identify mechanisms of compounds by clustering them in the learned embedding space. We can see several red squares at the diagonal of this heat map corresponding to compounds that have similar mechanism of action. We also observed that for the mechanisms we identified through this phenotypic clustering, the compounds annotated with those mechanisms are strongly similar to the knockout phenotype of their targets. For example, the representation you get from the cells when you perturb them with mTOR inhibitors is strongly similar to the one that you get when you knock out the gene mTOR. Let's look at the top plot on the right. The background distribution, represented in blue, is the cosine similarity of all compounds in our library to the mTOR knockout. And each mTOR inhibitor similarity to mTOR knockout is represented by a line of a different color. You can see that most lines are on the right tail of the background distribution, which means that mTOR inhibitors are more strongly similar to the knockout of mTOR than most of the other hundreds of thousands of compounds in our library. And at the bottom plot, you can see a similar trend for MEPK inhibitors. Predicting mechanisms for compounds also gives us the ability to de-risk them for adverse outcomes.
We've now seen that representations of small molecules and gene knockouts are functionally meaningful and can be related to each other. One immediate outcome is that we can identify compounds that match or oppose phenotypes of specific gene knockouts, rapidly providing hit compounds for phenotypic states of interest. This is a massive leap forward from single target screens of compound libraries because of two fundamental advantages. The first is speed. Any gene phenotype can be immediately queried as a drug screen, allowing you to quickly see if any biological hypothesis can result in compound hits. Second is the set of additional endpoints you can employ when each compound is in the context of other compound and gene phenotypes. These other relationships can tell you a lot about selectivity over other genes or avoiding similarity to unwanted mechanisms. As an example, let's look at the discovery effort for the oncology target MYC. MYC is a famous oncogene with driving alterations in the majority of all tumors. It's been the target of fierce drug discovery efforts for decades, but the discovery of MYC drugs has struggled in large part due to its simple structure and the lack of traditional binding pockets. In this case, we're using the phenotypic representation of MYC knockout to identify compounds that make cells look like they've lost MYC, whether or not they are direct binders. Shown in this heat map, we've identified several series of compounds that achieve similarity to the MYC knockout state. To confirm these findings, we followed up these hits to confirm their inhibition of MYC signaling via cellular assays such as luciferase reporter or MYC protein degradation. These compounds are starting points, but key to using any technology to enable drug discovery from early chemical matter is the ability to identify small improvements in potency as a chemical series progresses. For that, we'll use a separate program where we've progressed a compound series through a significant amount of chemical changes to build out the structure-activity relationship, or SAR. Here, we're relying on the platform to provide us with a clean, stable read on compound potency and magnitude so that we can see small changes among analogs. To do this, we can take the similarity of a compound to a desired phenotypic state as a function of dose, resulting in a traditional sigmoid curve common to evaluating a compound's performance in an assay, shown on the left. In the table on the right, you can see that an initial active compound in orange increases in potency and magnitude as chemical modifications are made, which is key to advancing early-stage hits into more drug-like molecules.
Hi, I'm Imran Haque, VP of Data Science at Recursion. I lead our data science department, members of whom work on everything from the assays and data pipelines that go into our maps of biology, developing the machine learning methods, building our representation spaces, and building the computational biology and chemistry tools that power search in our maps to drive therapeutic discovery. Let's zoom back out from individual gene-gene and structure-activity relationships. What has been the real impact of having built this meaningful representation, and where do we go from here? One capability enabled by a model that can embed general rather than just specific biology is that it can be rapidly pointed at new areas of interest. You may have heard of this disease called COVID-19. Early last year, we conducted screens both for infection by SARS-CoV-2 as well as against the cytokine storm that characterizes late-stage severe COVID-19. While we did not initiate our own clinical trials based on these results, we shared our results prospectively with the scientific community, and several of the compounds that we profiled were taken into trials by others. Of the compounds that went into large-scale trials, we predicted trial results correctly in 6 out of 7, and we know the reasons why we missed on 1. Furthermore, we released all the data from these screens to the public at rxrx.ai and at covid19.rxrx.ai to support future research. Looking at discovery more broadly, Recursion first deployed inferential search, the process of using these maps of biology that relate perturbations in our unified representation space, internally in July 2020 in oncology, and in early 2021, we transitioned nearly all new internal discovery efforts to use it. Based on progress achieved to date, Recursion has demonstrated that a program can advance from inference to in vivo validation in less than 6 months. The impact that inferential search in a unified representation space can make on accelerating drug discovery has already manifested beyond our internal programs. Just last week, we not only announced an expansion of our partnership with Bayer to apply inferential search to accelerate our collaborative search for drugs in fibrotic diseases, but also a new partnership with Roche and Genentech to apply inferential search to build up to 40 programs in key areas of neuroscience and in an oncology indication, as well as to collaborate on new machine learning methods on multimodal sequencing and imaging data to build highly granular maps of human cellular biology. So where do we go from here? The models discussed in this work were trained on selected subsets of the entirety of Recursion's data and already demonstrate meaningfulness across a broad range of biology. As of the end of the third quarter of 2021, to build its map of biology, Recursion had executed about 95 million phenomics experiments across a broad range of biological contexts, with a complete data universe spanning over 11 petabytes. We think that this dataset could serve as a backbone to build foundation models, capturing a broad range of phenomena in and applications of biology.
Hi, I'm Nathan Lazar. I'm one of the directors of data science at Recursion, here to just wrap up the talk with some key takeaways. So we showed you that Recursion's platform captures this high-dimensional phenotypic readout in multiple different human cell types at a massive scale. We've done genome-wide arrayed CRISPR knockouts and combined that with over 700,000 compound perturbations into the same embedding space. And what you can do with that, once you have these in the same space, is cluster these high-dimensional phenotypes, and that demonstrates utility by recapitulating known pathways of genes and known drug mechanisms in a diverse set of mechanisms of action in different biological functions. In addition to supporting known biology, we can interrogate frontiers of novel and classically undruggable space, for instance, the mix story that we told earlier. These same learned representations and phenotypes can be used to direct search through chemical space for discovery and structure activity optimization of new medicines. Just want to thank all the team that made this work possible, from the engineering side to people working in the lab. It's a great team to work with. If you have more questions, please come to our poster and check out our preprint that we published last year, detailing some of these methods as well as some of our work on COVID-19. We're also hiring both at our main office in Salt Lake City as well as in Canada, both in Toronto and Montreal. And you can go to recursion.com/careers to learn more about that. Thanks.